Hello students, welcome back to Goodness Pharma.
Today's practical is identification of carbohydrate from the marketed glucon-D sample. So we are taking the glucon-D and we are going to mix the 2 spatula of glucon-D in the distilled water. Mix it with the distilled water and stir well so that the sample is completely soluble. So the sugar sample is ready. This we are going to take for all the tests. Take 2 ml of sugar solution into the test tube. Now add the molish reagent 5 drops into the sugar solution in our test tube and mix it well. Now we are adding the H2SO4, concentrated H2SO4 by tilting the test tube from the side of test tube. So here we get the violet purple color ring at the junction of two liquid. So this is observation for the molish test, violet purple color ring at the junction of two liquid. This is general identification test for all the carbohydrates. Now the second test is for the reducing sugar identification. We are taking the Benedict reagent 5 ml into our sugar solution in the test tube. Now for the Benedict test, we have to heat the solution, Benedict reagent plus our sugar solution. Here the Benedict reagent, which is in the blue color, is turned into the brown color. The brick red PPT is observed in the Benedict test and it represents the reducing sugar. If the sugar given sample is reducing sugar, then the test will be positive, means the brown color PPT, brick red color PPT. And if the sugar solution is non-reducing sugar, then it will remain as it is, colorless. This is the observation of the Benedict test. Brick red PPT is observed at the bottom of the test tube. Take 2 ml of Barford reagent in the test tube and add sugar solution 5 drops. To the Barford reagent, add 1 ml of sugar solution. And boil over the flame for 30 seconds. The observation is the brick red PPT is observed for the Barford test and it represents the monosaccharide. If the given sample is monosaccharide, that is we have taken the glucon-D sample, that is glucose, so it is the monosaccharide, so test is positive for Barford test. This is brick red PPT observation for the Barford test. Next test is for the identification of carbohydrate, which is aldose or ketose. So for that purpose, we have to take the Selivanov reagent. 2.5 ml of Selivanov reagent is taken into the test tube and we add the sugar solution 1 ml. Add 1 ml of sugar solution into the Selivanov reagent and mix it well. Heat the solution for 1 minute and cool the solution. If the given carbohydrate is aldose, then the test will be negative, and if the given carbohydrate sample is ketose, then test will be positive. After 1 minute, the color of the solution remain as it is. It means that the given carbohydrate is not a ketose and it is the aldose. So we have taken the glucon-D sample which contain the glucose, and glucose in chemical structure it contains the aldehyde functional group, so it is the aldose. So the Selivanov test is negative for the aldose compound and positive for the ketose compound. For the final confirmation of the glucose, we take the ozone test. It means the crystals are formed. For that purpose, we have taken 5 ml of sugar solution in our test tube. Now we are going to add the phenylhydrazine hydrochloride. Now half spatula of phenylhydrazine hydrochloride we are going to take in our test tube, which contain 5 ml of sugar solution. Then add double the number of phenylhydrazine hydrochloride sodium acetate crystal. If we are taking half spatula of phenylhydrazine hydrochloride, then we have to take 1 spatula of sodium acetate crystal in our test tube. Then mix all the solution well and add 10 drops of glacial acetic acid. Add 10 drops of glacial acetic acid into the test tube. Add 10 drops of glacial acetic acid and keep the test tube in the boiling water bath for next 20 minutes. Now keep the test tube in the boiling water bath for 20 minutes. After 20 minutes, we observe the crystals that we call ozone, and from that ozone, we confirm the sample by their shape and color. After 20 minutes, we observe the crystals, and we are taking this on the slide for the observation on microscope at 10x. So take on the slide, adjust the slide on the microscope at 10x and observe the crystal. In 10x, these types of crystals are observed. These are glucose zone because these are having